Hello everyone. So the next topic which we will be discussing is about the interface. In these presentations, in these slides, we will be discussing that what are the three phases of interface, what are their functions. Then through a diagrammatic representation, we will understand the cell cycle and how the process of interface goes and for how much duration it will proliferate in the human cell culture. So we will be discussing this in the slides. So let's start with the first slide of introduction. It is the stage of life cycle of a cell where the cell grows and DNA is being replicated. Or you can say that it is the time duration during which the cell is preparing for division by undergoing both the cell growth and the DNA replication. There is a cell growth and DNA replication. DNA replication is in case of the S phase. We will be discussing the three phases where we will understand that what is happening in the interphase zone. So these are the three main phases of interphase. The first is G1 phase, which is also called as first gap phase. Then it is the S phase, which is also called as a synthesis phase. Then comes the second gap phase, which is also called as the G2 phase. So these are the three kinds of phases which are being mentioned in the interphase, or the interphase has been subdivided into these phases. So we will be discussing each one of them in detail. So first we will start with the first gap phase. So before starting with the first gap phase, we will see the how a cell cycle is been running in the cell basically. So here this is the G1 phase where the cell growth occurs. This is the part of the interface only. Then comes the S phase, that is where the DNA synthesis takes place. Then comes the G2 phase where the cell growth takes place. So that's why in the definition of interphase in the introduction slide it was written that it occurs in, it is basically related to the cell growth and DNA replication. So here DNA synthesis or DNA replication is being taken place in the S phase and the cell growth is being taken place in the G1 and the G2 phase. After the G2 phase there comes the mitosis or mitotic phase, where this is the part where the mitosis takes place and this is the part where the cytokinesis takes place. Cytokinesis here means the division of the cytoplasm. So word cyto, it depicts the cytoplasm and kinesis is used for the division. So here the division of the cytoplasm is being taken place. So the formation of two daughter nuclei, uh, the whole of this combines to form the two daughter nuclei. So this phase is called as the mitotic phase. So our main focus is not on this. In this presentation we will be focusing more on the interphase. So in case of G1 phase, it is the period between the end of the M phase. Here the M phase is ending and the start of the DNA replication is being depicted in this particular diagram. Then comes the S phase and it is the period during which the DNA synthesis occurs or DNA replication occurs. Then comes the G2 phase, which is the gap period following the DNA replication and proceeding towards the initiation of the M phase. So this is between the DNA replication phase and the initiation of the M phase. So this is how a diagram is being represented and the interface is being depicted, that this is the part of the interface, that G1 phase, S phase and G2 phase are the parts of interface. So now we will be discussing the first phase of interface, the first major phase of interface, that is the G1 phase, that is also called as the cell growth phase. So G1 phase, it is the period between the end of the M phase and the start of the DNA replication. So it is the first stage of interphase because it is called the G1 phase. Also because it is called a G1 phase because when we view it in a microscope, there is a very little change which is visible in the a G1 phase. So that is why it is the first phase. And the cells, they are quite active at the biochemical level. The cells are mostly biochemically active at this level. Then the cells grows and accumulates the building blocks of chromosomal DNA and the associated proteins, as well as the sufficient energy reserves to complete the task of replicating each chromosome in the nucleus. So the cells grow and they accumulate the building blocks of chromosomal DNA. And this phase particularly lasts for 11 hours for a typically proliferating human cell in the culture. So when we proliferate human cell in the culture, the G1 phase specially shows that it will take 11 hours to proliferate. So this was about the G1 phase. One more thing which has to be keep in mind, that approximately in a cell cycle, the 95% of the cell cycle is spent in the interphase. So the 95% is spent in the interphase, then comes the M phase, and all the phases comes after that. But 95% of the cell cycle is being denoted to the interphase. And interphase, it is called the resting phase, but here it is basically the most active phase, in which the cells are preparing for the division, for both the cell growth and the DNA replication in a very orderly manner. So these 2 points have to be keep in mind, that 95% is being depicted to the is being given to the interphase in the cell cycle. Then we will, after the G1 phase comes the synthesis phase, in which the DNA replication or DNA synthesis is being taken place. So it takes the longest because of the complexity of the genetic material which is being duplicated. So here the DNA replication is being take place. So there is a genetic material which is involved. So the interphase will take the longest here. The synthesis phase of the interphase will take the longest here. And in S phase particularly, DNA replication results in the formation of identical pairs of DNA molecules. So there is a replication, so there will be an identical pair of DNA molecules, cystochromatids, that are firmly attached to the centromeric regions. So the centromere is being duplicated during the S phase. So there is a replication, so the duplication will occur. So centromere is being duplicated during the S phase. And the two centromeres will give rise to the mitotic spindle. This we will be covering in all the other slides in detail, that through a diagrammatic representation, that what all is happening in the centromere, what is centromere and how it is being duplicated. So at this point we just have to understand that DNA replication takes place here, and there is a centromere which is being duplicated. So at the center of each animal cell, the centromere of the animal cells are associated with the rod-like objects like centrioles, which are at right angles to each other. So the centrioles basically helps to organize the cell division. So centrioles are the main thing which is responsible for the cell division. So centrioles are not present in the centromeres of other eukaryotic species such as plants and most fungi. So if we talk about S phase, it takes about 8 hours in a human cell culture to proliferate. So we discussed about the G1 phase, that it takes about 11 hours, but in the S phase it takes about the 8 hours. The next phase which we will be covering is the G2 phase, which comes just after the DNA replication. So this is the gap period which is following the DNA replication and preceding the initiation of the M phase. So what is happening in this phase is, in the G2 phase, the cell replenishes its energy and stores and synthesis proteins which is necessary for chromosome manipulation. So some cell organelles are duplicated and the cytoskeleton is being dismantled, and they provide the resources for the mitotic phase, because just after the G2 phase it is the initiation of the M phase, which is being depicted in the diagram also. So there are some additional cell growth which takes place during the G2 phase. So there is a additional cell growth which has been taken place. So the final preparations for the mitotic phase must be completed before the cell is able to enter the first stage of mitosis. So before entering the mitosis phase, that is just after the G2 phase, the final preparations, that is what all is needed for the mitosis phase, it has been completed almost in the G2 phase. And if we talk about the duration, that in a human cell culture, how much time will it take to proliferate? It takes almost 4 hours to proliferate. So this is the reference. The